I would now like to shift to the analytic part of this presentation, and in view of what we've been discussing about the CQAs that we just came up with, kind of formulate some goals for analytics. So one obviously would be to determine the total amount of glycans, or the so-called glycan occupancy on the molecules, and especially here for sialic acid. We would like to know the ratio of the sialylated versus the acylated glycans, and less important would be to know, for example, what are the characteristics of the sialylated structures. So how do they really look like? Where are they located? And for example, also what kind of linkages are present in the proteins? Because that's something that you usually know when you know what kind of cell line you're working with. Maybe there's an exception. There are approaches where people try to change 2-3 linkage that you usually have in CHO cell lines to the more human linkage, which is the 2-6. So if you do some genetic engineering to achieve that, that may be of interest for you. So if we look at the approaches that one can take to analyze glycans and sialylated glycans, then there are 2 ways to do it. This is a very complicated matter, so I'm not gonna go into too much detail. So there are 2 main approaches: the glycomics approach and the glycoproteomic approach. The glycomic approach is based on the analysis of glycans that have been released from the protein. You can either do this with PNGASF to release the N glycans. For the O glycans, you have to use a different protocol, which does a reductive beta elimination to get rid of the O glycans and afterwards analyze them. The glycoproteomic approach works either on entire sialoglycoproteins or on peptides, and both approaches are really complicated, especially if you want to preserve the sialic acids, because they are rather sensitive towards the next steps which are following, which are usually the labeling with fluorophore and cleanup, etc. So it's important not to lose the sialic acid, and that applies for both of these approaches. Maybe what is important to highlight here, or if you compare these 2 approaches, the glycomics approach has the big advantage that the workflows are largely standardized, which is not the case for the glycoproteomic approach, which is a lot more exploratory and needs more expert knowledge, and also a lot more complicated data analysis is required for this. So that's just a short, brief overview of the different approaches that you can take to analyzing glycans and sialylated glycans. So in all, what you end up is having, quite a challenging situation, because the glycan mixes, especially with high degrees of sialylation, is hard to resolve with chromatography, for example. And the sialylated glycans are charged as opposed to other glycans which are neutral, and that's just making the separation even more difficult. And that's also the reason why sometimes people have one process which is only focusing on analyzing charged glycans, and another method that only looks at neutral glycans, and afterwards the results of both methods are combined to give it the complete picture. And as I said, also the labeling and derivatization, which is involved in these methods, can lead to significant loss of the sialic acids. That's something which is kind of shown here. So in acidic conditions, high temperatures, and over longer reaction times that are needed for this derivatization, you can lose some of the sialic acid in the process, which means that you really have to make sure that your protocols are properly set up and really preserve the sialic acids in order to get precise results. So to summarize that, we are always dealing with complex methods with multiple sample prep steps. We have to deal with different methods and protocols for N-glycans and O-glycans. Those both require experts and are also rather costly equipment to run it. And in the end, it's still quite a low throughput method, which limits the number of samples that can be measured, for example, in a cell line development process or a bioprocess development. So, of course, the question arises, and that's the question that we try to answer: are there alternatives that can potentially measure high sample numbers with an accuracy which is sufficient to assess product quality early in CLD and bioprocess development? And maybe is there a technology out there that doesn't require really a lot of analytical expertise and possibly also works on non-purified samples? So that leads me to potential solutions which involve screening assays to overcome some of these limitations of the traditional analytical methods. So when we looked into this subject, we came across lectins, and you may have heard about lectins already. So lectins are plant-derived proteins, which are a great tool to quickly assess the overall glycosylation on proteins. They specifically bind to sugar residues, so on the proteins, so to the certain kind of sugar residues on the proteins. What is great is that the sialic acids can be measured on O-glycans and on N-glycans with essentially the same protocol, just using a different kind of lectins. The lectins bind only the accessible glycans on the glycoprotein, which what we believe is the important part of the information. The protocols are rather simple, so there's no digestion involved in labeling, etc. And the data analysis is also rather easy. So, for example, if you look at a microarray that uses lectins, then you see triplicates of lectins spots here somewhere, and the intensities of these spots tell you how much lectin has bound, and this signal correlates with the total number of glycans of a certain type in this given sample. So one nice feature of these lectin assays, it's not only a sialic acid assay, but this assay can be complemented by, for example, information about galactosylation and hymenosylation using these the appropriate lectins as well. So what you get with lectins is you get signals that indicate the presence of the different sugar residues on your glycoprotein. The disadvantages of lectins are that the lectins are not very strong binders to the sugars. So we're talking about high micromolar affinities here. So one needs really a proper optimization of the binding conditions for the individual lectins. And the other point, which is important to mention here, is that lectins also don't provide structural information. So if you get a signal for the galactose, for example, this includes this is like a sum parameter for all the galactose-related fibers in the sample. So how do we at PAIA use the lectins? As I mentioned very quickly in the beginning, what we do is bead-based assays in a high throughput manner. So what we do is we use capture beads with a functionalization on it, which capture either the sample or the lectin that we have in the mix. So what we do is we bring all these three components together in the wells of our special microplates. That's what's happening here. Afterwards, we're shaking the plate at room temperature on a shaker. And once the assay is finished, the beads settle down. They are separated from the unbound lectin, and this lets us directly determine how much of the lectin has bound to the sample and gives us a number that corresponds to the amount of glycan on in the sample.